Hello, welcome to Adaptive Immunity 4. So this is kind of a synthesis. We're putting stuff together. So you've learned the major players, macrophages, neutrophils. These are all cells I'm mentioning, by the way. Macrophages, neutrophils, B cells, T cells, natural killer cells. And then let's look at how they all work together to keep you healthy. Chapter 16. So this is the cytokine network. I think I may have shown this diagram in the past, but it's just how the white blood cells talk to each other. So for example, here's a macrophage, here's a B cell, here's a T cell, here's a mast cell, a neutrophil, a basophil, an eosinophil. And you can see all these little purple and green rectangles with letters in them. Those are all the cytokines, or the names of the cytokines. So for example, all these IL molecules or cytokines are interleukins. Inter is between leukins or leukocytes, which are white blood cells. IFN is interferon gamma and interferon beta. There's a bunch of interferons. And TNF is tumor necrosis factor. But you can see that there are a lot of molecules, or cytokines in this case, that the immune cells use to communicate with each other. Cytokines are soluble. That means they can dissolve in the fluid portion of the plasma. They're regulatory proteins. That means they regulate how these cells do their job. And they act as intercellular signaling molecules between white blood cells. They are secreted by various white blood cells. So all these white blood cells, macrophages, T cells, B cells, etc., secrete them. And in order for another cell to respond to a secreted TNF alpha, for example, that cell has to have a TNF alpha receptor. So cytokines are secreted by various white blood cells. They affect diverse cells in both the adaptive and innate immune system. So remember, our major cells in the adaptive system are B and T cells, and then our major cells in the innate are macrophages and neutrophils and natural killer cells. So cytotoxic T cells, or TC cells, do not respond to an antigen unless they are first signaled to respond to that antigen using cytokines. So there's 5 groups of cytokines. There's the interleukins, there's the interferons, which are mostly antiviral proteins. There's growth factors, or GFs. There's this, they direct the stem cells in the red bone marrow to divide. TNFs are made by these immune cells. They kill tumor cells. That's how they were first discovered. And then chemokines, or chemotactic cytokines, that help cells move to the site of infection. So this is just a B and T cell review. So this is in your book. This is kind of funny, so I put it in here. We have examined the cast of characters. So like the immune system is like this complicated play, and we have to look at the cast of characters in the immune system. And the characters aren't always cells, they're just things. It could be molecules or cells. So the cast of characters include antigens and their epitopes. So here's an epitope. This big purple thing could be part of a bacteria or an antigen. T cells with their T cell receptors, so they can bind things here. B cells and their B cell receptors, so here's where they bind things. Cytokines, we just looked at those, TNF alpha, interleukins, etc. And then actors that disrupt the play. T and B cells, if they went through their maturation process and should have died off but didn't, and they can recognize autoantigens or self-antigens, and then they can destroy those cells that have the autoantigens on them, then that would be a disaster, and that would be an autoimmune disease. All All right, we've talked about MHCs, major histocompatibility complex proteins. So here's a normal cell, and it's got its MHC marker in it. Check it out. There's nothing in there, though. There's nothing in there. B and T cells ignore it. If you come over here, here is, this looks like it might be like a macrophage probably, and it's got its MHC protein here, this green thing, and then these little dots in there are foreign antigens, aka non-self antigens that were processed inside the cell. That now the T cell can recognize those foreign antigens presented in an MHC complex protein. MHC proteins are also called HLA, human leukocyte antigen cells, are proteins. They're found on the surface of most animal cells, and we're animals, and they present epitopes or slash antigens to T cells. The real function of major histocompatibility complex molecules is to present epitopes slash antigens to T cells. If you are a T cell, with your T cell receptor, you can only recognize an epitope or an antigen that is presented on an MHC. And here's class 1. All nucleated cells have class 1 MHCs. That's where the antigen slash epitope binds. If you're an antigen presenting cell, like a B cell or a macrophage or a dendritic cell, you can present your antigens in an MHC2 molecule. MHC protein, they get their name because of their role in tissue rejection, major histocompatibility complex. Are you compatible with other tissues? Each antigen presenting cell has about 100,000 different MHCs. So now this is interesting because we've been saying in our T cells and our B cells that they have, if you are a single T cell or a single B cell, you have identical T cell receptors all over your plasma membrane. If you're a B cell, you have identical B cell receptors. But in this case, with these MHCs, if you are a cell, specifically an antigen presenting cell, you could have about 100,000 different MHCs sticking out of your cytoplasmic membrane. And we've been talking about what APCs include: B cells, macrophages, dendritic cells. Okay, these are cartoons for T-dependent exogenous and endogenous antigen processing. So this is where you would present your antigen to a T cell, and then the T cell would do its job. So it's T-dependent. So let's look at this first column. This is processing of T-dependent endogenous antigens. So here you are. Here's some cells in your body. Okay. And here's the nucleus of the cells in your body. And what happens is inside the cytoplasm of a cell, these cells, normal cells, are constantly chopping up old proteins. And in that process, if you are a bacteria, you get chopped up and you had a protein on you, that protein would get chopped up as well. So that's why I say here, these could be, these green balls here, this could be a self polypeptide or a non-self polypeptide. And then that polypeptide would have self or non-self epitopes. And then what happens is these epitopes can get inside the endoplasmic reticulum. You see them going into a little port right there. And then on the inside of the ER, or endoplasmic reticulum, you have these MHC1 proteins. This blue thing here. And you can see, if you look at the tip, it's got a different shape than this MHC1 protein. And so that means they can bind to different epitopes or antigens. Okay. It says here the MHC1 protein is in the membrane of the ER. And in the membrane of the ER, you can load your epitopes onto these MHC1 proteins. So then then you can, this thing here will pinch off and become a vesicle. And inside you've got all your MHC1 proteins with, check it out, they're different epitopes on each one of those MHC proteins. And then you can, if you're a vesicle, you can travel out here to the cytoplasmic membrane, and you can fuse with the cytoplasmic membrane, and then when this thing flattens out, your MHC1 protein will be presenting an epitope to the outside of the cell. Now, if a cytotoxic T cell comes by and with its T cell receptor recognizes this MHC1 epitope as foreign, then the cytotoxic T cell will kill this cell. And what that means is this original cell here was infected. But if this green triangle, say, is just a self-antigen, then the cytotoxic T cell with its receptor would not recognize that green triangle because it would be a self epitope, and you don't want your cytotoxic T cells to recognize self epitopes or antigens. Okay, so that's T-dependent endogenous antigens. So endogenous as opposed to exogenous. So T-dependent exogenous antigen processing. So here we have probably a macrophage. It's doing phagocytosis, taking in the bacteria or the pathogen. It's got these little green antigens on it. Goes into the phagosome and the lysosome, and then the phagosome and lysosome fuse into a phagolysosome. And then, so here's this guy. He got all cut up in a phagolysosome, and here's a vesicle that has these MHC2 proteins in them, and you can see, look, they all have different shapes, so they're all going to recognize different epitopes. And then the vesicles fuse, and you load your epitope antigen onto your MHC2 complex, and then it's the same thing that comes up here to the cytoplasmic membrane. They get this gets flattened out to the outside, and you've got your MHC2 protein displaying an epitope. And if this is a non-self epitope or a foreign epitope, then T helper cell will recognize that as foreign and do its thing. So class 1 MHC molecules are essential. So we're over here, class 1 MHC molecules are essential for presentation of endogenous antigens. Class 2 MHC molecules are essential for processing of exogenous antigens. So it's kind of weird, but I mean, both of these bacteria go into a cell, but in endogenous antigen, that means it came in and infected the cell. Exogenous antigen, that means the bacteria was engulfed by the macrophage. This thing didn't infect the macrophage, it was engulfed, actively engulfed by the macrophage. Okay. So we'll look at this a little more detail, endogenous antigen processing. So before an MHC protein can display the epitopes, so here's the epitopes, they must be processed. The epitopes have to be processed, and that happens in the endoplasmic reticulum. So if you are a pathogenic non-self epitope, that would include proteins produced by viruses or pathogens, and you break those antigenic proteins into really small pieces of about 8 to 12 amino acids, and the epitopes are included in that. And then we talked about this, the epitopes are bound to the MHC1 here in the endoplasmic reticulum. They're packaged into vesicles here by the Golgi apparatus, and then they're inserted into the plasma membrane. If these are pathogenic epitopes, you trigger a cytotoxic T cell and it kills you because you got infected. Exogenous antigen processing. So an antigen presenting cell internalizes a pathogen, and this pathogen's here. It's got non-self epitopes on it. It breaks down the pathogen and produces all the epitopes in the phagolysosome. And then it fuses with the vesicle with the MHC2 proteins. And then you get a vesicle that goes to the plasma membrane of the cell. And if a T helper cell recognizes this epitope in an MHC2 complex, and that'll trigger the activation of the helper T cell. And this is a scanning electron micrograph of an actual dendritic cell. So that's kind of cool. So, summary so far, we've looked at all the organs, the cells, signaling molecules. We've looked at clonal deletion. We've looked at antigen processing. We know adaptive immunity is specific. That means that the T cells and B cells with their T cell receptors and B cell receptors are looking for specific complementary antigens or epitopes. We know adaptive immunity involves clones of B and T cells, and those B and T cells should not be responsive to self-antigens. And we also have talked about how the immune responses are inducible against foreign, aka non-self antigens. You don't want the immune response to be induced against self-antigens. So again, here's our T cell receptor, and here's our class 1 MHC protein. So remember, if this cell, any nucleated cell in your body, got infected and chopped up the pathogen, put one of the pathogen's epitopes right here, and then this T cell receptor, bound to a T cell, recognized it. So let's, I wish I could have drawn these cartoons upside down, but anyway, recognizes it, then that activates the T cell. If it's cytotoxic T cell, then it will destroy the infected cell. The T cell mediated immune response, which I just talked about, is not at the site of infection. It's usually in a lymph node. So this T cell receptor recognition of an MHC1 protein usually is happening in the lymph node. And here's our lymph node. And it usually happens right here in these germinal centers. That's where all the action is. Okay, so this is cartoon's activation of cytotoxic T cells with no help from T helper cells. This is rare. This doesn't happen often. This is like T independent activation of cytotoxic T cells. So let's check it out. Here's our cytotoxic T cell down here. Here's our infected cell. So clearly virus got in, and the virus inserted its DNA here. And then the viral DNA made viral RNA, made viral peptides. And through some really cool molecular biology, those viral peptides got linked up to a complementary MHC1 protein. And now this infected cell presented this viral peptide, or this epitope, in a class 1 MHC molecule. The cytotoxic T came along, recognized the class 1 MHC molecule holding this viral peptide, and the T cell receptor, when it recognized it, the CD8 co-receptor bound to it too. And now it doesn't show it in the cartoon, but now this cytotoxic T, when all this synapse happens, now the cytotoxic T can kill the infected cell. So if you are an antigen presenting cell like a dendritic cell, it says here, the infected dendritic cell could go to a nearby lymph node and find a buttload of these cytotoxic Ts, and it can present, this infected antigen presenting cell can present viral epitopes in an MHC1 protein. At least one cytotoxic T cell in the lymph node will have a complementary receptor to that presented viral peptide. That cytotoxic T cell will bind to the infected cell. It forms this immunological synapse. And then CD8, this co-receptor, stabilizes the whole thing. And then cytotoxic T cells kill the infected cells with chemicals. And there's our lymph node again. All this is happening in the lymph node. Okay, so let's take a look at this cartoon. This is activation of cytotoxic T cells with the help of helper T cells. So this would be T-dependent activation of cytotoxic T cells. That means that these cytotoxic T cells need the help of a helper cell, T helper cell. So here's our hero, the macrophage. Here's our antigen, so a virus or a bacteria going into the cytoplasm of the macrophage. And remember it's a nucleated cell, so it's got this class 1 MHC. The antigen on the pathogen gets processed, so it gets cut up. And then the epitope, or the antigen part of the foreign invader, gets put into a class 1 MHC molecule and then presented here on the surface of the macrophage. Now the cytotoxic T cell can come along with its T cell receptor, and it can recognize, if it's got the right shape, cytotoxic T cell receptor can recognize that processed antigen in the class 1 MHC. Okay, now this gets a little tricky. So there you go, and you can see that the cytotoxic T cell has formed this synapse, this immunological synapse here, but it doesn't say that it's activated yet. It needs the help of a helper T cell. So, so all this stuff here that happened between the macrophage and the cytotoxic T cell is this here. But now this macrophage also can present a piece of that invader as an epitope or antigen in a class 2 MHC here. Now the helper T cell can, with its T cell receptor, can recognize that MHC2 antigen epitope complex, and it doesn't show it, but the helper T cell would secrete cytokines, maybe like an interleukin, maybe like IL-1, and it would fully activate this cytotoxic T cell. And now, once you are an activated cytotoxic T cell, you can go out in the world and you can find infected cells that are infected with this specific antigen epitope presented in an MHC1 molecule in an infected cell. And now the cytotoxic T cell can, pour all its granzyme and perforins and kill that infected cell. That's the help of that's activation of a cytotoxic T cell with the help of a helper T cell. So when an interaction between a cytotoxic T cell and an antigen presenting cell is not strong, like we saw here, the helper T cell can help out by secreting cytokines and fully activating the cytotoxic T cell. And again, all of this is happening here in these germinal centers in the lymph node. Okay, now let's look at activation of cytotoxic T cells and helper T cells. So it's a different cartoon. This is a dendritic cell. So the dendritic cell was out in your skin. It found a pathogen, chopped it up, and it presented an epitope from that pathogen in its MHC2 protein to a helper T cell. The dendritic cell also chopped up that same bacteria and presented another epitope from that pathogen in a Th1 cell. But whether you're a dendritic cell, so you can present in an MHC2, you can present in an MHC1, so it shows it here. And then the T helper cell, its T helper cell receptor recognized the epitope in an MHC2. The cytotoxic T cell receptor recognized an epitope in MHC1. And this just shows a bigger picture of it. So it shows the CD8 molecule with the cytotoxic T cell. And it doesn't show in this cartoon, but there'd be a CD4 with the helper T cell. So that's antigen presentation. You're a dendritic cell, you're presenting the antigen to helper T and a cytotoxic T. Then, after that happens, you can see the dendritic cell secretes interleukin 12 to fully, so when this synapse happens, that tells the dendritic cell to release IL-12, which activates the TH cell into a Th1 helper cell. And then when this synapse happens, the inactive cytotoxic T cell with an IL-2 receptor on it comes down here, and this cytotoxic T cell has an IL-2 receptor, so it can respond to IL-2 secreted from the helper cell, specifically the Th1 cell. When that happens, now the cytotoxic T is fully activated. It can form a clone. Can form a clone of active cytotoxic T cells. It also forms a bunch of memory T cells. So if you get sick again with this same antigen, the memory cells will differentiate into active T cells right away. And then I like this. This is kind of funny. To really fully stimulate and activate yourself, if you are a cytotoxic T cell, you secrete IL-2, and then you bind IL-2, and you get full activation. And now you get this clone of active cytotoxic T cells. They go out in the world, they leave the lymph node, and they use these T cell receptors to look for antigens, these original antigens that were up here at the beginning of the cartoon. And then they'll kill the infected cells using perforins and granzyme. Co-stimulation between white blood cells requires epitopes and cytokines. So co-stimulation, you have an epitope here in an MHC2, you have an epitope here in an MHC1, and these cytokines. So co-stimulation requires these two things, and you're stimulating your cytotoxic T so that it can become fully activated and thus go out and kill cells that are infected with the original pathogen. During clonal expansion, these cytotoxic T cells are immediately active and memory cells are inactive. Your memory cells can last for months or years. You could be immune to this original pathogen for years. Response to specific pathogens can improve with subsequent exposure. So that means if you are originally exposed to a specific pathogen, when you're exposed again, then the memory cells will kick in, and you might not even know that you're sick. In primary exposure, so that's the first round of exposure to a pathogen, clonal selection occurs, producing these memory T cells for future responses. I think that's it. Nope. One couple more. Okay, so this is just another cartoon showing kind of what we talked about It's this kind of a little confusing, but this tan box is in the red bone marrow. This tan box is in the thymus. And then if you're in any of this box here, you are in a lymphoid tissue or organ. And then if you're out here in this color panel, then you're in the blood plasma. But it's basically the same thing. Here's an antigen presenting cell presenting an antigen in a class 2 molecule to a helper T cell. Here's an antigen presenting cell presenting an antigen or an epitope from that same foreign invader, but it's presenting it to a cytotoxic T cell or a CD8 positive T cell. But then you get formation of the clones and the memory cells. And then the clones, these guys all go out into the blood plasma. They travel all over the body looking for infected cells. When they kill, when the cytotoxic T cells kill, so if this is the plasma membrane of an infected cell, the cytotoxic T's will punch into the membrane these molecules called perforins, and then that'll make a hole inside. To get inside the cell, they dump in this enzyme called granzyme, and that granzyme initiates apoptosis, and then the cell undergoes suicide. And then we just said this, perforin molecules make a channel, granzymes enter the cell, and the infected cell is killed apoptotically. So it's really important that your body carefully regulates this cell-mediated immunity. So T cell is cell-mediated immunity, so that your body doesn't respond to self-antigens. And when this happens, as you know, it's an autoimmune disease. This is just another way to kill. If you are a cytotoxic T cell and you recognize that you recognize a virally infected cell, you can use this other pathway to kill. The end.